can you all now see my the single cell portal screen? Great. Single cell portal. You can get there by just typing in this URL, singlecell.broad-institute.org/single_cell. You can also just Google it. That's how I normally do it. When you're at this homepage, I'm just going to first take you through a little bit of a tour of the things you might notice here. First, there's the search bar down here. So as I mentioned, it's easy to run a simple text-based search using this search bar. And I'll demonstrate how to do that in a moment, but I just want to draw your attention to the fact that you can search across for individual studies or through this gene search tool. Then at the top here on the right, there's a useful button for help and resources, which will link you to some of our documentation, as well as information on how to email us if you have any questions. And here's more documentation on using APIs and scripts to interact with SCP. Just to the right of that is this button to create a study. This is the first step in, you have to first sign in. So I will do that now, but you can see that I could also have signed in by clicking this button. Creating a study is basically just the first step in the process of uploading your data. At this point, you just name it, and you can point it towards an existing workspace on Terra or create a new one. And you can choose here whether you want to add it to a brand or a collection, such as the BICCN Anatomy and Morphology Project, or just have it be a free-floating study. Okay, but I'm not going to create a study today. Instead, I'm going to start out by showing you some other features. So one last thing I want to point out on this homepage before we dig into some actual data is this button right here, which allows you to browse through the study collections. So I'm just going to briefly sign up, because otherwise I'll show you all the private collections as well. If I click on this button now, I can see the collections that exist so far and are publicly viewable. So here's the BICCN Anatomy and Morphology Project, for example. And here you can see that I just have access. Is basically just a portal within the portal that only shows you the studies that have been specifically added to this project. And I can do the same sorts of search functions as I click on the home page, but that will be constrained to this collection. Okay, so I'm going to quickly sign back in just to make sure I can show you all of the features that are accessible to me. I'm going to start out by showing you how to search through these studies. So as I said, there's a simple search option where you just search for a term that might appear in a study description. So I'm going to, for example, look for astrocytes, and this just returns any study with astrocyte in the description. There's also an option to use this more advanced faceted search. So for example, I could constrain my results to studies with data in astrocyte cell types, and then search for something like lung cancer. And here I get a much smaller, more constrained set of data that are easier to sort through. This time I'm just going to go to an individual study, which has been set up just for us, so that we can dig into some data. So when I first click on this study, or any study, I'll see a summary of that study like this. This is the information in the summary is totally up to whoever set up the study or who owns the study, but it often includes a brief overview, a link to a publication if one exists, an abstract, and sometimes there are links also to additional resources. For example, you could link out to visualizations for other portals as well, especially if they're, often these are a really good complement to what visualizations SCP offers. You could also include an image in this description if you want, such as a graphical abstract or a slide image that you is relevant to the data. Then the next tab over is the Explore tab. And this is the real meat of the data exploration capabilities of SCP. So what I have here is just sort of the default plot. So this is a two-dimensional dimensionality reduction. So as I mentioned, the data's original shape is something like number of cells by number of genes. And then that sort of full high dimensional data have been passed through a dimensionality reduction algorithm, in this case a UMAP, to project it into 2D space where it's easier to spot patterns. So every point here corresponds to a cell, and all these points are colored according to the label that's selected in this annotation dropdown. So right now it's selected to display cell type, and you can see from this legend what each of these cell types is. So to make it a little bit easier to identify each of these cell types on this plot, you can just click and then it'll disappear, and click again, and that same group of cells will reappear. It's just sort of like it's waving at you. And so that will help me to sort of easily identify this red set. But it's also, at this point, it's also informative to see things like how many cells belong to each set of each kind of cells. For example, there are a lot of glutamatergic neurons, but not very many macrophages, and also to understand sort of how these are arranged relative to each other. This suggests that the genetic expression profiles for macrophages is pretty different than that for astrocytes. Okay, so now, of course, you can also change this annotation. Instead of cell type, I could color these same data according to donor ID, and notice that everything that colored blue here, which came from one donor, is quite distinct from everything that was in green and red, which came from two other donors. But maybe I have some specific question that bears on that distinction. Okay, so now at this point, you might be wondering what differentiates, I'm just going to go back to the original annotation here, cell type. You might be wondering, okay, so these red and brown cells, for example, seem to belong to two different cell types, but they seem pretty similar to each other in that they're close in this plot. What genetic expression patterns differentiate these two cell types? To try to get at that, you can search for an individual gene up here in this gene search bar. So say I want to look into the P10 gene, I can search for that. And now you'll see a few new visualizations. So the first one I want to draw your attention to is this plot on the left, which is basically the same plot as what's on the right, except now each cell is colored according to the measured P10 expression. And from this, you can see that P10 is kind of expressed throughout the data. There's no one cluster where it's strongest, although you might notice that this sort of serpentine cluster of oligodendrocytes has relatively lower expression. But perhaps this helps to, you might search around for some genes that do distinguish between these cells or these clusters a little bit more. Another way that you can look at this is by clicking over to this distribution tab, which has appeared. And this plots violin plots for, again, P10 expression within each individual cell type. This can be a little bit easier to eyeball sometimes than the gap plot. You can also play around here with whether you're looking at a box plot or a violin plot and look for outliers, et cetera. There's a lot to explore here. Once you've sort of looked at a P10 expression and are thinking you might be interested in some other genes, you might want to check out this ideogram viewer at the top here. This viewer sort of highlights the gene that you searched for, and then also shows other genes that might be related based on the literature. These are either interacting or paralogous genes, and you can hover over any of these to get a little bit more information. If one of these piques your interest, like if I wanted to look into PTK2, you can just click on that and it will re-render this plot to instead plot PTK2 expression. And here you can see, interestingly, it does seem like this purple cluster of glutamatergic neurons has seemingly at least higher expression of PTK2 than the rest of the data. And you know, a whole here understanding all of these patterns. The last thing that I want to draw your attention to here is that if you found a subset of the data that seems particularly intriguing to you, you can use this create annotation option. This is something you have to be logged in to do, which is why I logged in so I could show it to you. But you basically just click this. You could name that annotation something more descriptive than that, hopefully. And then let's say I want to zero in on these macrophage cells. To do that, I just sort of draw a lasso around those cells. I can label these macrophage, not macrophage. Create that annotation. And what happens is that will save that this segmentation of the data. And once it's been saved, which will take a second, you'll be able to see it in this annotations dropdown where you go up to your user menu and then go to my annotations and it'll be there. And then you could download that or share it with collaborators if you want to see what they think of it as well. Okay. Then the last thing on this tab here is if at this point you're intrigued enough by the data that you want to play around with it yourself, you could go around to this, along to this where you can choose individual files to download, or you can click the bulk download button, which will basically just give you a command that you can use in any command line, any terminal window to download all the data at once. One thing to note here is that not all studies will make it possible for you to download your data. Some studies will be private, or they will say something like the data will be released at some future date. And in that case, you might have to email the authors that are associated with that study in order to ask them nicely for their data. So that was a tour through the different ways in which you might explore the data in an individual study. I'm also going to show you how to search for individual genes across studies. So if you go back to the homepage like I just did and click on this search genes option and enter a gene name or multiple gene names, let's go with P10 again. Then you'll get some results that basically you'll get a summary of that gene's expression, measured expression across any study that has data on that gene. So for example, here, this one's a little bit more interesting. You see a violin plot that tells you the distribution of P10 expression measured in each of these, in this case, it's clusters of the data, though you could also select some other segmentation of the data, such as the subcluster. This helps you to just sort of quickly eyeball which studies prominently feature a given gene. You can also, as I mentioned, search for multiple genes at once. And when you do that, you will get a slightly different sort of summary. So let's go back down to this study here. So this is a dot plot that tells you, that helps you sort of compare expression of these two genes across different subsets of the data. So for example, here I can see that both of these genes that I searched for are highly expressed in the CA2 cluster of the data, but they are differentially expressed in the GABAergic cluster of the data. And so if I'm looking for differential expression that pulls apart these two genes, this might be an interesting study for me to target. You can also view these plots a little bit differently. You can plot another violin plot where this time you're just averaging across two genes, presumably two genes that you expect to be relatively related to each other. And this will tell you the averaged expression distribution for those genes. Okay, so now I've shown you a few different ways to explore the data. So I'm just going to wrap up in the last few minutes by telling you a little bit of information about how to control access to your study. I think this is really key ability that SCP gives you. So I've already created a study of my own. So I'm going to go to my studies. This will take just a second to render because I'm the product manager on SCP, I have access to all the studies, which is kind of a lot. But I'm basically going to open up an individual study that I made. It's just a dummy study. It doesn't have any data. And I'm going to show you how you can share access to your study with individual people, including with anonymous reviewers. So I basically am just going to open up the study, which at this point, as I said, has no data, but this is something that can be done also when it has data, and go over to the settings button. Here, I can change things like the data release date, which I mentioned before. This allows you to make it possible for people to download your data sometime in the future, but not yet. You can also control whether the study is public or private, meaning whether it will show up in search results. And you can share the study with individual people. You can either allow them to view the study, even if it is private, or to edit the study, in which case they could change the data on Terra to sync it with this study. So this is helpful if you have a collaboration going, especially if like one of you is a wet lab scientist and one of you is a computational scientist. You can pass results back and forth. But then the one thing that I think is really useful is this reviewer access. So this makes it possible for you to share access to a study that's still private with an anonymous reviewer so that they can view your data without actually downloading the data. So you would basically just click over to say access is enabled, but if you wanted to disable it at any point, you would just click no. And then an expiration date, meaning when you want the reviewer to no longer have access to your study. And then doing this, once you've made these changes and then clicked update study settings, you can see these reviewer access credentials. So there's a URL and a PIN. You would send these both to your journal editor, essentially, so that they could pass it on to the reviewers without breaking their identity. And so what they would do is once they got this URL, they would click, I should have opened that. Yeah, well, anyway, they would use this URL to access a sort of access points. And at this point, they'd have to enter in their PIN. And once they've entered in the PIN, they'll be able to access your data, but only for 4 hours. This is just to make sure that they're not likely to like erroneously share your data with someone else or something like that. If they get kicked out after 4 hours and they still want to go back and see it, they can always just re-enter the PIN. Yeah, so that I took you through a lot of different features there. I know I was hoping to give you a sense of the many different ways that SCP can support your full research life cycle. But I know I just threw a lot of information at you. So if there are any questions, I'd be happy to answer them.
Layla, I have a question. I realize this might be a silly one, but is there any way to save some of the graphs that you generate in there? Did you show that or?
Yeah, I did not show that, but there is. Yeah, that's a great question. I'm just gonna select this study at random. So if I wanted to save this graph, I can click this camera icon, which will just download the graph automatically as a PNG file.
Okay, very cool. And can you do the same for kind of the dot plots and things that you were showing too? Is that like if you're searching it, just across genes? Yeah, like searching across studies or something, and you wanted to save one of those dot plot figures.
I haven't tried to do that, but it might be. I think it has a slightly different control, but you can go to save image. Oh. Yeah, it's a slightly different way of doing it.
Oh, cool.
But that is possible as well. Great question.
Would it be also good to maybe talk a little about the URL and how it can save some of the state of single cell portal?
Right. Yeah. You mean like the URL connected with this particular visualization? Yep. So say when I originally landed on this page, I'm gonna go back to, so, okay, this is a good demonstration actually. If you've changed something about the visualization, as I have here by searching for a couple of genes, and I want to go back to the original view, I can just click reset view down here. And then, if I wanted to share this exact visualization, say I wanted to change the annotation a little bit, and I wanted to make sure that I was sharing this visualization with a collaborator, I could go over here to get link, and then just copies that link to my clipboard. And then if I were to send that link to someone, they would see the moment of truth. They would see exactly that visualization. So thanks for that reminder, Tim.
I was wondering how many UMaps could you have in here, and how many metadata can you have plotted on the UMaps?
That's a great question. I'm not actually sure of the strict limits there. I mean, like you can see, it depends upon the study how many different UMAP options there are, for example, under clustering here. But you can see if I choose a different one, I actually get a different plot because it's a different projection of the data. Part of the power of SCP is that researchers can choose the different kinds of representations of their data that they want to share. Similarly, sometimes there are just a couple of annotation types, but sometimes they're up to like 20. And it just depends upon what data are supplied. Well, feel free to shoot me an email anytime if you have questions, and I hope you guys get to play around a little bit in SCP.